it over to you.
Thank you very much for the introduction. I hope everybody can see my slides. So what I'm trying to do today is to give you an overview of highly pathogenic avian H5N1 influenza virus. You hear a lot about that in the news. And I wanted to give some background and give an overview of the current situation. And if you have time, I'm also going to talk a little bit about the current situation with SARS-CoV-2 and COVID-19 vaccines. But let me just start with my disclosures. I think the most important one is that Mount Sinai is developing SARS-CoV-2 vaccines that have been, and IP for that has been filed. And Mount Sinai is also working on spinning out a company, CastleVax, to commercialize these SARS-CoV-2 vaccines. And I'm a co-founder of that company, and I'm on the scientific advisory board. And that will become relevant in the end because I was asked to give a little overview of where we are with that vaccine development. But I'm going to start out with H5N1 and with influenza virus in general. And so, as I said, I'll start really with a general introduction to bring everybody to the same page. So we're all familiar with flu, with influenza virus, right? Influenza viruses are part of the Orthomyxoviridae family. And we actually have 4 types. We have influenza A, B, C, and D. Most people are familiar with influenza A and B because they are clinically relevant, right? They cause disease in humans. Influenza C also causes disease in humans, but it's usually common colds. And influenza D only circulates in cloven-hoofed animals, in cows, in sheep, and so on and so forth. The virus itself is an enveloped virus. That it has a lipid envelope that's host cell derived. You see that here on the left side, this yellow layer. On the inside of the virion is an RNA genome, and that genome actually comes in genomic segments. You can think about that like chromosomes, right? There's 8 of these genomic segments for influenza A and B, and that's one of the problems that we have with influenza, as you will see, because different viruses can exchange these chromosomes or these genomic segments. And these 8 genomic segments code for 2 spike proteins, the hemagglutinin and the neuraminidase. HA, or hemagglutinin, is the receptor binding protein, and NA, or neuraminidase, is the receptor destroying enzyme. And when you hear H1N1, for example, that refers to the subtypes of the glycoproteins that the virus has. An H1N1 virus has an H1 hemagglutinin and an N1 neuraminidase. But there's many of these subtypes, and we'll get back to that. And then another genomic segment encodes for the matrix proteins. Then we have another one that codes for the non-structural protein. And then we have 4 more segments that code for the polymerase complex and the nuclear protein. What do we know about influenza in terms of the disease? First of all, influenza viruses have a very wide host range. They can infect basically everything from humans to alligators. In humans, they cause a spectrum of mild to severe respiratory disease, everything from asymptomatic disease to fatal disease. Basically, we see epidemics with influenza. That's our seasonal flu, and they are caused by influenza A and B viruses. And then occasionally we have pandemics, and they are caused only by influenza A viruses. In terms of epidemic or seasonal influenza, we know that worldwide we have between 3 and 5 million severe cases every year, and somewhere between 300,000 and 650,000 deaths. In the US, the CDC estimates that influenza kills between 3,000 and 50,000 people every year. That's seasonal influenza, and the strongest example for pandemic influenza is the H1N1 pandemic of 1918 that claimed approximately 40 million lives, but the estimates go from 20 to 100 million. If we look in other mammals, we see basically the same. Influenza viruses cause a spectrum of disease, mostly a respiratory disease in other mammals. That includes ferrets, pigs, even marine mammals like seals, whales. Influenza sits in horses. It can infect cats, dogs, and so on and so forth. But the main host of influenza viruses are avian species, birds. And here we have mainly infections in the GI tract, and very often those are asymptomatic infections. The birds might not even be sick. Sometimes we get systemic infections, and we'll talk about that when we talk about H5. And with these systemic infections, typically then there's severe disease, and the birds can die of these systemic infections relatively quickly. Just as a recap in terms of human influenza and pandemics, if you look at the last 100 years, we had a number of pandemics. We had the 1918 pandemic, which was caused by H5N1. And this virus was introduced in the human population, caused the pandemic, and then kept circulating and became a seasonal virus. And because, of course, people acquire immunity over time and mount neutralizing antibody responses, there was pressure on the virus. And so the virus evolved over time to escape these antibody responses. These changes over time that we see with influenza viruses are what we call antigenic drift. And then in 1957, we had another pandemic with a different subtype, H2N2. And that's what we call an antigenic shift if the subtype changes. Interestingly enough, this H2N2 virus kicked out H5N1. We don't understand these dynamics really, but this is what happened. And then H2N2 was drifting and circulating until 1968, when we had another pandemic, another antigenic shift. In this case, it was an H3N2 virus. This again eliminated H2N2, and H3N2 is still circulating in humans. It's actually very diversified and causes a lot of issues. 1977, H5N1 came back, a very similar virus to the one that went extinct in 1957. This then circulated until 2009. And in 2009, another H5N1 virus, distinct from this one, jumped from pigs into humans, and that's our 2009 H5N1 influenza pandemic. And so the situation is relatively complex in humans. We have now H5N1 circulating and changing all the time, H3N2, and then also influenza B. And we have no clue what the next subtype will be that causes a pandemic. When we think about viruses that can cause pandemics, that can jump from animals into humans and start a new pandemic, we have to think about what these viruses need in order to break the species barrier and become human-to-human transmissible, right? First, again, transmissibility among humans is important because if the virus can spread among humans, it can cause a pandemic. And here, the receptor binding of the virus comes into play. Typically, these avian influenza viruses bind to sialic acid that has an alpha-2-3 linkage. And the human viruses, typically the seasonal influenza viruses, typically bind to sialic acid that has an alpha-2-6 linkage. In our system, in the human respiratory tract, in the upper respiratory tract, we have mostly alpha-2-6 linkage. Deep down in the lung, we also have alpha-2-3 linked sialic acid, but that explains why the virus needs to bind to 2-6 linked sialic acid, because it needs to be able to replicate well in the upper respiratory tract to kind of basically spread from humans to humans. But typically, avian influenza viruses don't have that. There's also a question about pH stability that needs to be right for human-to-human transmission. And then the hemagglutinin protein needs to be activated by proteases. And so we have to have a sequence in the hemagglutinin that can be cleaved by human proteases in the airways. Otherwise, the virus cannot infect more cells. In addition to that, there's often adaptation to the mammalian host replication machinery, right? So the influenza polymerases have to interact with the cellular machinery of our cells. And typically, avian influenza viruses, avian polymerases are adapted to avian cells. And so in order to replicate well in human cells, the virus needs to mutate. And one well-known mutation is in one of the polymerase subunits, and it's this E627K mutation. Sometimes we hear about that mutation now in the news when a new human infection with H5N1 is found and the virus is sequenced. In addition to that, we have restriction factors against avian influenza viruses. For example, MXA, that's a protein that is expressed in human cells. And human seasonal influenza viruses circumvent this easily, but avian influenza viruses are restricted by it. And typically, it's not the case that an avian influenza virus just jumps into humans and then starts to spread from humans to humans. Typically, what you have is a reassortment event, right? I talked about this genomic segments earlier, and so when you have two different influenza viruses, they have their 8 genomic segments each, right? But if they infect the same cell, they can actually exchange genomic segments. And so if that happens, for example, with an avian influenza virus and a human influenza virus, and they infect, for example, in a pig or in a human, the same cells, they can exchange genomic material. And the outcome then could be a virus that now has the hemagglutinin and maybe the neuraminidase from the avian influenza virus, to which we don't have a lot of immunity, but the replication machinery of a human virus. And that would allow the virus to replicate very well in our cells. And that's a problematic situation. And in fact, the pandemic of 1957 was caused by one of these reassortants. The 68 pandemic was caused by a reassortant, and the 2009 pandemic was also caused by a reassortant. It's very likely that this also happened in 1918, but we just don't have enough information about the situation back then. And there's very little information about the genomic makeup of the virus from 1918 known. And so it's very likely that the next pandemic is also caused by a reassortant virus, and that's something we have to keep in mind. And the problem is that in the animal reservoir, we have a lot of diversity. We have all these different subtypes of influenza viruses. So on the left side, you see a phylogenetic tree of hemagglutinins, right? And on the right side of neuraminidases. And you can see how much diversity we have here. For hemagglutinin, we have for influenza A, group 1 hemagglutinins and group 2 hemagglutinins. And then the influenza B hemagglutinins are a little bit separate from that. Looks very similar for the neuraminidase. And what I marked here with these red stars are subtypes of hemagglutinins that either are currently circulating in humans or that have caused pandemics before, like H2. And then the blue stars indicate subtypes that have caused human infections, zoonotic human infections, often with relatively severe consequences, like with H5 and H7. This is all to say there's a lot of diversity out there, and all of these subtypes could hypothetically jump into humans and cause a pandemic. And what makes this more problematic is also that all these subtypes circulate in wild birds, but also in all kinds of other animals, marine mammals, domesticated animals, even pets. And basically, there is constant reassortment of these viruses, right? And so there's a lot of diversity out there, and we don't know what the pandemic potential of all these viruses is. Another important point is, of course, all these viruses might circulate in animals, but as long as we don't interact with these animals, they can't infect us. So interaction of humans with the reservoir species is also something that is a risk factor for a new pandemic. And there's actually a lot of these interactions, of course. You see this picture on the left here, this kid basically kissing a pig. This is there because we had situations in swine fairs, in agricultural fairs in the Midwest, where people were interacting with pigs that had swine influenza, and then people got infected with swine influenza that way, basically. And what you can see here is a timeline of recorded zoonotic infections where different subtypes from birds and from other animals jumped into humans. And you can see that this basically happens constantly. And here on the right side, I have more recent cases. There's actually a lot going on. We had H7N2 that jumped into cats and into a human. We had avian swine H1N1 in Asia that jumped into humans. H3N8, that's an avian influenza virus that recently jumped into humans in China. Those viruses typically only infect a few people and don't spread, but they cause infections. And then, of course, we'll talk about H5N1 in a minute. And H2N2, this virus that caused a pandemic in 1957, is also still around. And it's actually being detected every now and then at wet markets, including in New York City. There's wet markets in Queens, and H2N2 has been detected there in chickens. So let's talk a little bit about H5N1. So H5 is one of the hemagglutinin subtypes of influenza A. It was isolated for the first time in 1959 in Scotland from chickens. We have two different versions of H5N1. We have a low pathogenic avian influenza H5N1, and that's abbreviated LPAI. And then we have highly pathogenic avian influenza H5N1, and that's abbreviated HPAI. And basically, the difference between these two strains in terms of disease is huge, but in terms of sequence, the difference is very small. I told you earlier that the hemagglutinin needs to be activated, right? So the hemagglutinin protein needs to be cleaved in two parts by host proteases. And the low pathogenic avian influenza viruses have a monobasic cleavage site. So basically, this hemagglutinin can be activated by different airway or gut proteases, but not in other cells in the body. And highly pathogenic avian influenza viruses have a polybasic cleavage site. You see this RKRR here. And this hemagglutinin can be activated by furin-like proteases, and they are present in all cells. And that means that this virus basically can spread throughout the body, at least in birds. And that makes it highly pathogenic. And this is just an example how this looks in birds. This case, chickens. You can see chickens die of highly pathogenic H5N1 relatively quickly, and we have virus replication all over the bird, right? So in all kinds of tissues, it's almost like Ebola for humans. And here on the right side, you see Kaplan-Meier curves of white leghorn chickens that got infected with different high BEF H5N1 viruses, and they really die within 2 to 4 days, right? So relatively quickly. But for a long time, it was known that this virus existed. It was a problem for the poultry industry, but nobody really thought that the virus could actually infect humans. And that changed in 1997. So 18 human atypical influenza cases were detected in Hong Kong in 1997. Actually, 7 of these people died. And they found out quickly that this was caused by a highly pathogenic H5N1 virus. So this was circulating there in chickens. People went to wet markets, interacted with the maybe bought chickens there, and then got infected. And some people got severely sick and unfortunately died. And this is H5 that's associated with the A goose guangdong lineage. That's a lineage that emerged in China. H5 has many different lineages, and that developed into a high BEF phenotype and caused this issue in Hong Kong in 1997. And it turned out when people studied this virus, these highly pathogenic avian influenza viruses, that they are actually pretty devastating in mammals too. And here on the left side, you can see an experiment, data from an experiment in ferrets, where people took one of these high BEF H5 strains and then infected ferrets, and you can see weight loss here. So the weight is declining very quickly. And then they made a version of the virus that doesn't have this polybasic cleavage site in the hemagglutinin. And in that case, the ferrets also get infected, but they don't die. They have very mild disease. And you can also see the difference here on the right side. This here is the virulent version with the polybasic cleavage site. And the virus in the ferrets even spreads to the nervous system and the brain. And that's how these animals unfortunately succumb to infection. The virus without this cleavage site doesn't do that. I think the positive thing here was that the virus, these H5 viruses that were isolated in Hong Kong and also later, don't transmit well in mammals. You can test this in the ferret model. If you, for example, infect ferrets with human H3 viruses, and then you co-house them with naive ferrets, you can see these are virus titers. They're spread from the inoculated ferrets to the contact ferrets. And that's also what happens in humans, right? H3N2 is a seasonal human influenza virus. But if you do the same with these H5 highly pathogenic viruses, the virus replicates in the inoculated ferrets, but it doesn't actually spread to the contact ferrets. And this is true in an aerosol transmission model where the animals don't directly touch each other, but it's also true in a contact transmission model where they can freely interact. And I think for a long time, this was really saving us from an H5 pandemic, because the virus is just not able to spread in mammals very well. So this was detected in 1997, and then for a few years, there were no detections and there were no human cases, not even avian cases. But then in 2003, the virus came back, and back in the day, people panicked about that because all of a sudden you saw a lot of cases, specifically in Southeast Asia, and people thought that the next pandemic would come right away and it would be H5N1. And what happened next was that with migratory birds, right? Birds migrate via over whole continents. There's specific flyways that they use. The virus was spread in this case mostly by mallard ducks and by geese from Southeast Asia to basically the Middle East, to Europe, and to Africa. And so some of these wild bird species get infected, but the virus doesn't really make them too sick. And so that's why they can spread the virus well. In chickens, it's usually highly pathogenic. And so we saw introduction of these H5N1 viruses in Europe, in Africa, and in the Middle East. And while this was happening, the virus was also diversifying. And this is just an overview of the different lineages of these high-path H5 viruses that we have right now. And it changed a lot over time. And I'll get back to that in a minute. What also happened was the virus started to reassort, right? Initially, it was an H5N1 virus, and then over time, it reassorted with other avian influenza viruses that had other neuraminidase subtypes. And so we saw H5N2 emerging, H5N6, H5N8. And at some point, these viruses actually made it to North America for the first time ever. And that was in 2014, 2015. And we didn't have human infections back then, but a lot of poultry operations were affected, and actually egg prices went up during that year. But the virus wasn't very fit, and it actually disappeared from the wild bird population in North America within a year. In the meantime, a lot of things were happening in Europe and in Asia and in the Middle East. And actually, with the spread of these H5N1 viruses, more and more human cases were reported. Again, zoonotic infections, so people got exposed to birds and then got infected and then got sick, not human-to-human transmission. And here is basically a summary of the cases between 2003 and 2023. And I think what's important here is, if you look at the case numbers, there's a total of 882 cases that were reported during that time, and 461 of those died. And that gives you a case fatality rate of about 50%. So it seems that this virus can be very deadly in humans. But there's a lot of discussion about that, because of course, the case fatality rate only takes into account the people that that are cases, right? People who are sick that then get tested for this. And it's often people who are severely sick already. But there have also been serological studies in specifically Southeast Asia in populations that specifically work with poultry, have contact to birds. And it turns out that often the prevalence, and this is done by serology, the prevalence for these viruses is actually quite high, right? So a good proportion of the population seem to have been infected and seroconverted, but haven't gotten sick. So the infection fatality rate on the other side, which takes into account also asymptomatic infections, might be quite low for H5N1. But there is still an active discussion about that. I said there was a lot of diversification, and currently there's two different clades that we're dealing with. And I'm sorry about the nomenclature here. I didn't come up with that. I think it's it's very confusing. But currently we have clade 2.3.2.1 E that's circulating in Southeast Asia. And when you hear about human H5 cases in Cambodia or Vietnam, they're caused by this clade. And the clade that we are hearing about in North America right now and in Europe, for example, is clade 2.3.4.4b. And that is really currently very, very widespread. And that's also the issue in the dairy cows that you hear about in the media. And so this clade 2.3.4.4b, again, is an H5N1 virus. And that seems to be very fit. And it started to really expand in 2020 during the pandemic in wild birds in Europe. And then in December 2021, this virus arrived in North America, again via wild birds, likely via flyways over Iceland. And then in 2022, this virus started to spread in North America and later made it to South America, and it's now also present in Antarctica. And what's special here is that we see a lot of mammals that get infected, often dead-end infections, right? Predatory animals that feed on carcasses of birds that had died with H5N1, and then the mammals get infected and then they die. But we see that at a very high rate. And in addition to that, there were also cases of mammal-to-mammal transmission in fur farms in Spain and in Finland, in mink and in foxes. And a lot of infections actually in marine mammals in Chile and Argentina. And the suspicion there is also that there was mammal-to-mammal transmission. And we had, so far up to that point, a number of human cases. Most of them were actually mild, except for one in Central America and one in South America that were severe cases. And then we started to see actually more and more cases also in the US. And I'll talk a little bit later about the actual numbers that we have right now. What happened in March of this year was something that was very, very unexpected. It was reported that H5N1 had made it into dairy cattle in a farm in Texas. I'm saying this is very unexpected because typically cattle doesn't get infected with influenza A. There's basically no reports of influenza A infection in cows before we had this outbreak with H5N1. And so the way they noticed this is cows in these dairy farms that were affected gave less and less milk. And so that means that they have health issues, and then people started to investigate, and it turned out they had H5N1. And after a lot of sequences were looked at and a lot of viruses from different dairy farms were sequenced, it turned out that this was likely a single introduction from a bird to a cow. And then the virus started to spread from cows to cows. And the interesting part here is that in the cows, it doesn't seem to be a respiratory disease and respiratory spread, but it actually seems to be spreading via milking equipment. Just to give you an update of the situation there, this is from the USDA. In total, we had infections, H5 outbreaks in more than 800 dairy farms in the US. In more than half of the dairy farms in California are currently affected. So this is really massive. And for a long time, people were struggling to understand how this is spread until, was really investigated how this could spread, specifically without a lot of respiratory disease. But it turned out that there's a lot of virus that's secreted in milk. And so basically, the cows get infected through contaminated milking equipment. The virus replicates in the mammary glands to very high titers. And actually, a lot of infectious virus is secreted into milk, partially up to 10 to the 9th plaque-forming unit. So that's really, really high. It's hard to get those titers in cell culture if you grow the virus in a laboratory. And actually, from that, basically milk secretion, the milking equipment gets contaminated and then spreads from cow to cow. And then I didn't know that before this outbreak, but there's actually a lot of transport of cows from one farm to another. And the idea is that that is how the virus started to spread throughout the US in this dairy cattle. And what also happens is now that, for example, cats that live on these dairy farms also come in contact with the milk. They probably drink the raw milk, get infected, and die of the infection. And there has also been spread from these dairy farms now to poultry operations and back into wild birds. So it's actually a pretty complex situation. And with that, we also have seen more and more human cases in the US. Right now, we're up to 58 cases. Most of these cases were relatively mild so far. There are some exceptions, and most of them could actually be linked to exposure to infected cattle or exposure to infected birds. But there's 3 exceptions. There's one case in Missouri that was actually a more severe case where it wasn't clear how the patient got infected with H5N1. There was no clear interaction with birds or with cattle or infected milk or contaminated milk. And then there is now one more case in Canada and one more case in California where we see that. And that's, of course, a situation where people get worried, because if you don't have a clear exposure, then it could also be already human-to-human transmission. And if you see human-to-human transmission, that means that the virus can spread from humans to humans, and that would indicate that we're getting very close to a new pandemic. The problem that I also see here is that, of course, there's a lot of people working on these dairy cattle farms, and they get exposed to the virus, they might get infected. And now we're going into influenza season, and these people might get infected with seasonal influenza viruses, and then might still go to work and might get then co-infected with H5N1, and that can lead to these reassortants. And again, these reassortant viruses might then be fitter in humans. What's also interesting, we had recently one case in California where there was potential spread from or potential infection via consumption of raw milk that was contaminated with H5N1. So raw milk consumption is really not something that I would recommend. Another important point here is that's often overlooked. When we think about avian influenza viruses, we often think about rural environments, we think about farms, we think about birds. But actually, there's a huge interface between humans and animals in urban areas too. If you think about New York City, there's actually a lot of green spaces. There's a lot of birds here, right? Not in Central Park, but not just Central Park, everywhere. There's a lot of other animals that we interact in the city too. Within the city too, right? And so there's actually also a certain risk that we get exposed to avian influenza viruses in New York City or other urban environments. And I just wanted to highlight, we started, this was actually started in 2019, an initiative to do surveillance here in the city. And we're doing that in a science outreach and community science project where we actually go with high school students and collect bird feces in New York parks, and then basically try to extract RNA from these samples and see if we can find avian influenza viruses and other viruses that circulate in these birds. And this is done together with New York City Parks and the Wild Bird Fund, which is a bird rehab facility on the west side, and then of course Mount Sinai, and also with BioBus, which is a community science organization. And of course, in the end, we found H5N1 in the city. We actually recently, with the students who are doing that work, published a paper on detection of highly pathogenic H5N1 in the city. And we have since then, this was the first 6 isolates that we reported. We have since then found a bunch of more isolates. So just because we're in a city doesn't mean we are safe from avian influenza. There are a lot of countermeasures for H5N1 or influenza in general that exist. So the situation is very different than the situation early in 2020 with COVID-19. There's a lot of approved influenza virus therapeutics, neuraminidase inhibitors, ion channel inhibitors that are not used currently in clinical practice, but they're still available. And then also endonuclease inhibitors, and they also work well against H5N1. So we already have drugs to treat these infections. We also have vaccines and vaccine candidates for H5N1. H5N1 vaccines have been developed as early as 1997. The problem is that they're not super immunogenic, and they usually need to be given with an adjuvant. And then there is also heterologous prime boost regimens where live attenuated vaccines are given first, and then inactivated vaccines against H5 are given a few weeks later, and that typically enhances neutralizing antibodies. There's also basically development of mRNA vaccines right now for avian influenza viruses, specifically for H5N1, but it's unclear how well they work. Avian influenza virus vaccines for H7 and H10 have been tested with mRNA technology in the past, and they were not very immunogenic. So maybe the traditional vaccines in this case actually work better. And the good thing is also that the USDA is now starting to test H5 vaccines in dairy cattle, and that might actually help to stop that outbreak. So I'm just going to check the time quickly. I'm going to give a very brief overview of the current SARS-CoV-2 situation as well, just because I think it's interesting, and I guess the expectation is that the cases will increase over the winter again. And so I'm going to switch from influenza virus to coronaviruses. Just a brief introduction. We look at the numbers from the pandemic. There's now estimates that COVID-19 actually caused approximately 27 million deaths globally. These estimates include data until 2023. So that's actually a pretty devastating number, a pretty high number. The official numbers are much lower, but of course the official numbers often didn't record all the deaths, specifically in low and middle income countries. So this is probably much closer to the truth. But a lot of things changed with COVID-19, of course, and this is illustrated nicely here by this data from the CDC. So what you can see here, basically in yellow, is weekly test positivity from CDC studies, and you can see this goes up and down depending on the waves of virus infections that came in, but it's still occurring, right? There's a lot of COVID around, sometimes less, sometimes more, but it didn't go away, right? What you see here in blue are weekly deaths. And I think that's the good news. Initially, we had a lot of mortality from COVID-19, but it seems to become lower and lower and lower. And this is, of course, because a lot of people got vaccinated, a lot of people have immunity. It's not because the virus becomes weaker, it's really because we have developed immunity. And this is the good news. It doesn't mean that the virus is not dangerous anymore, specifically if you have issues with your immune system. It could be still, or any other underlying disease, it could be still very, very problematic. And of course, there's still a lot of issues with long COVID. But I think we really transitioned from the pandemic situation to an endemic situation where we see much less severe damage done by the virus. Also, the virus, as you all know, changed a lot over time. Initially, we had what we now call the ancestral virus. Then, as you remember, in 2021, the first variants emerged, and then Delta came, which was a big problem. Then Omicron started to circulate in the beginning, in the end of 2021, beginning of 2022. And then there were different waves of different Omicron versions. XBB was dominating for quite some time. This is also a descendant of Omicron. And now we're in the era of JN1. Which is also a descendant of Omicron, basically. This is data from Mount Sinai Hospital that was provided by Harm van Bakel. They do a really good job at monitoring what's going on in the hospital samples, and you really see basically something similar here compared to worldwide circulation of these variants. This is from the CDC. This is the current subvariants that we see. So all the variants that you see here are JN1 subvariants. What's most prevalent right now is KP3.1.1 and a variant that's called XEC that seems to slowly increase over time. It's not really dominating yet, but it seems to basically push KP3.1.1 out slowly. And so what I wanted to highlight is this naming, the nomenclature for the variants can also get relatively confusing. But whenever you see an X, that means that we have a recombinant virus. And so I was talking earlier about reassortment for influenza viruses, when influenza viruses infect the same cell and then exchange genomic segments. Coronaviruses can do something similar. When they infect the same cell, they don't have genomic segments, right? They have only one genome strand. but recombination can occur. And so XEC, when you see this X for a variant, that means it's a recombinant. And so XEC is basically a recombinant of two JN.1 subvariants of KS.1.1 and KB3.3. And so XEC is actually not very different from the parental viruses or from KB3.1.1. It's just a few amino acids, but it seems to be a little bit fitter in the population compared to KB3.1.1. The antigenic change is relatively small, so it's still pretty close to the strains that we have in the vaccine. And because we had so much change with these viruses over time, as you all know, we had variant-specific vaccines that were introduced. In 2022, we had the bivalent vaccines with wild-type spike and then BA5 spike. And then last year, we had the XBB.1.5 vaccine, right? And so what the CDC is doing on a regular basis now, and actually I hope the report for the current vaccine is coming out soon, they look at vaccine effectiveness. So how well do these vaccines work, right? And so they studied that for XBB.1.5. And what you can see here is the vaccine effectiveness for the XBB.1.5 vaccine from last year. And what they see for these updated vaccines is an effectiveness that's somewhere between 45 and I would say 55%, which is actually pretty good. This is vaccine effectiveness. It's not vaccine efficacy, right? So this is very different from what was tested initially when the clinical trials were run, the phase 3 trials, where you have a placebo control group and then your vaccinated group. So you're comparing vaccinated individuals with people who have no immunity whatsoever. And here for this vaccine effectiveness, basically what is compared is people who have been recently vaccinated with people who are not recently vaccinated, but certainly also have some type of immunity against SARS-CoV-2, right? So if you get to 50, 60% here, it's actually not bad. So the vaccines, the updated vaccines are certainly working. There's another study that came out of Cleveland Clinic that looked, and specifically because we had a change in variant last year, from XBB to JN.1. So they looked at basically the era before JN.1, and they also see 40%, 42% vaccine effectiveness. But what they see when the variant changed, right, when the virus evolved, then the vaccine effectiveness went down. And this is basically a very good justification for updating the vaccine again. And now this year, we have a JN.1 subvariant in the vaccine. Depending on which vaccine you're using, the Pfizer and Moderna vaccines have the KB2 subvariant in the vaccine, while the Novavax is based on JN.1. But they should all pretty well match with what is circulating. So I think for this season, we're good, and we should see good vaccine effectiveness. And just the last 2 or 3 slides, was asked to give an overview of the vaccines that we are developing here at Mount Sinai for COVID-19. And they are based on a virus vector, a Newcastle disease virus. And this is work that I'm doing with Wayna Son and Adolfo Garcia Sastre and Peter Balesi also here in the department. The current COVID-19 vaccines that we are using are all injected, and that those injected vaccines give us pretty good systemic immunity, and systemic immunity can also protect the lung very well. But it isn't very good at inducing immune responses in the upper respiratory tract, right? And that's what you ultimately want if you want to protect yourself from infection, right? If you want to protect from disease, specifically severe disease, protecting the lung is important and sufficient. But if you want to protect from infection, you need strong immune responses in the upper respiratory tract. And this is something that potentially could be done with intranasal vaccination. And so this is something we're currently working on. And as I said, we're using a Newcastle disease virus vector for this. So this project goes back to the beginning of the pandemic, when we didn't know which vaccines would work. And it wasn't even clear if mRNA vaccines would work or if they could be produced in sufficient numbers, right? This had never been done before at large scale. And so we were thinking, okay, there's a lot of production capacity for influenza vaccines globally. Can we use that to make COVID-19 vaccines? And as you know, influenza vaccines are mostly produced in eggs. And so we were looking for a virus vector that would grow in eggs so we could use these facilities. And we came up with Newcastle disease virus. That's a virus that's infecting chickens. It's problematic in chickens. It doesn't really cause disease in humans, but because it causes disease in chickens, vaccines, attenuated vaccines have been developed. And so we used one of these vaccine strains and introduced the sequence for the spike protein of SARS-CoV-2 into that genome of Newcastle disease virus. And what you get when you do that is a Newcastle disease virus that has a lot of spike protein on the surface, so the SARS-CoV-2 spike. But when it infects a cell, it also expresses spike protein. And typically, it can't really replicate well in human cells, but it goes into the cell and forces the cell to express the spike. And so we made these viruses, and you can see here in these plots, in these STS pages, on the left side, the wild type version of the virus. So you see the different proteins in these virus preparations, the NDV proteins. And then here on the right side, you see preparation of this construct, which we call NDV hexapro S. And this has an additional band here, and this is the spike protein of SARS-CoV-2. And so basically, you can now produce these vaccines in embryonoided eggs in facilities that typically make flu vaccine. And like flu vaccine, you can either grow the virus and then inactivate it and purify it and inject it intramuscularly. So we're doing that in some projects. But you can also purify it and then use it as a live intranasal vaccine. And that would give you immunity in the upper respiratory tract. And just to give you an overview here where this stands in terms of development, we have initially worked with GPO and IVAC. Those are two vaccine producers in Thailand and Vietnam to develop an inactivated intramuscular version of the vaccine. And this is now licensed actually in Thailand. And it's still in development in Vietnam. And we have also worked with a company in Mexico, Avimex, and they use also an intramuscular version of the vaccine, and this is now also licensed in Mexico. But what we are really excited about is, and this is a development that was started here at Mount Sinai, also clinically, is an intranasal version of that vaccine. And so a phase 1 trial was completed here at Mount Sinai, and we have now gotten funding from BARDA, from the US government, to do a phase 2B trial, so actually already an efficacy trial in 10,000 people, and to really look if this vaccine, when you give it intranasally, can protect you from infection. And that trial will start in Q1 of 2025. And we're all very excited, and we hope that it's going to work, but we'll see and we'll have to wait and see what the data looks like. And so with that, I'm going to end this presentation. Maybe we still have time for questions, if there are any.
Thank you, Dr. Krammer. This is sort of an amazing journey, both some virology education for all of us, in addition to some zoology. And I think it's just fascinating, the connections between that's sort of going on all over the world between the infection in other species and how we all interact. Just reminding us how important the whole climate issues are in our work. I'm just wondering, if you could talk a little bit about some of your work with the flu virus and possibly, there's thoughts of combining COVID with flu virus and sort of what's on the horizon. And the other thing is, just given what you've said about the avian influenza virus, what's the role of primary care? I mean, you mentioned some interesting work on sort of basically doing some serologic studies. Is there a role that we could play in some of the data that you're collecting?
All right, let me start with the combination vaccines, right? So there are actually a number of vaccines in the clinical pipeline, actually very far, phase 3, that combine SARS-CoV-2 and influenza vaccines, right? This is on the one side done by companies that work with mRNA-based vaccines, but also Novavax, for example, has a recombinant protein vaccine that combines that. So far, none of those vaccines have been licensed, but they're very close. I think one of the worries are that people won't take those vaccines, right? The influenza vaccination rates in the US are relatively high, right? I think more than 60% of the population gets vaccinated. The COVID-19 vaccination rates are relatively low right now. And so the question really is, how big is that's a question that the companies are asking themselves, right? How big is the market for a combination vaccine? And of course, currently we have a recommendation from the CDC that the COVID-19 and SARS and influenza vaccines can actually be given at the same time on the same day, right? So that already makes it convenient. So we'll see how that develops, but basically the vaccines would be there from a development perspective. We are at Mount Sinai, we're taking a different path. We're actually trying to develop a universal influenza vaccine, so vaccines that would protect you independently of changes in the virus, right? Independently of this antigenic drift, independently of which subtype it is that's circulating. That is work that is in phase 1 trials right now. So this is going relatively slowly, but there is progress there as well. We're actually starting 4 clinical trials in 2025. But it will probably take until 2026 until we have results from these trials. But again, this is early clinical development. And then your last question was, you know, what could be your role? I think it would be important to, in the current situation in the US, to really keep your eyes open. If you get somebody with respiratory disease who had interactions with poultry, with wild birds, with dairy cattle, or who is known to consume raw milk, right? Keep in mind that this infection could be an H5N1 infection. I think the problem that we have right now is there is no easily accessible tests for H5N1 out there, right? So there are a number of diagnostics for influenza that will be positive when you have an H5N1 case, because they test for nucleoprotein, for example, and that's relatively conserved, but there's nothing specific. So you would have to go basically to a state lab, and then even if the state lab, if Wadsworth tests the sample positive for influenza, it would still need to be confirmed by the CDC. So it's relatively, there are relatively strong barriers right now for doing that if you have a suspected case, right? But I think it's really important to stay watchful, because at any moment in time, this virus could take off in the population and you could see more and more cases. And then, you know, basically the medical community would have to act very, very quickly. So it's really important to keep in mind that this is something that can happen, that these infections can happen, and that it's not something that, you know, wouldn't happen in New York City, for example. It can happen here too. So I think it's really important to stay on top of that and think about it if you have patients who could potentially be infected with H5N1.
Well, yeah, that's what's great about these presentations. It reminds us to ask these questions when we're presented with a clinical scenario that puts people at risk. That's helpful. One question that somebody asked is about the frequency of COVID vaccination. And are we sort of approaching an annual COVID vaccination like the flu?
Yeah, exactly. I think that is really where we are going, right? So influenza vaccines are given basically once a year because they are updated. And we also see that there's waning immunity, you know, after vaccination, after 6 months, immunity goes down. So it's not just the updated strains, it's also waning immunity. And I think we're seeing similar things with SARS-CoV-2, specifically for people who are at higher risk. The annual vaccination makes sense. But I think in general, what you're doing with these annual vaccinations when you have new variants, right? You're re-educating the immune system. You're basically telling the immune system, okay, this is how the virus looks now. This is basically a refresher course every year for the immune system. And I think from that perspective, it does make sense.
Well, as always, this is just education in so many different fields in medicine and healthcare, and again, zoology and virology. So we thank you, Dr. Krammer, and look forward to next year's talk. So
Thanks for the invitation.
of course, thanks everybody. Have a good holiday, and we'll see you in January. Take care.